Hi guys. All right, so this time we are going to talk about the this Mendelian genetics. So let me share screen first. Hang on a second. Let me open it up. Where is it? Where is it? Are you guys? Okay, let's do that. Let's do this. How do I open it? Okay. That might work. Nah, not that one. Not that one. How do I do it? Maybe that's this one. But I don't want slideshow. Okay. Hope that work. Oh man, don't slideshow. Don't slideshow. Maybe not. Okay. Okay. So this is Mendelian genetics. So we will talk about the very original idea about the genetics that was very first observed by George Mendel in about 18th century. So that was even before we know about the DNA and chromosome. And his finding actually still can be applied to the current, society. But after knowing the genetics more today, we not just confirm what George Mendel have found, but also we, explore beyond Mendelian's law. Okay, so we will talk about these two, Mendelian laws and beyond the Mendelian law. All right, so that's first one. So what's the George Mendelian's doing is that he plants, right? And he found that like mating two different plants will have different offsprings. So for example, that he has a tall plant and a short one. And he found that, okay, we got two tall and two short. So the chance is 50-50. And that one probably makes sense to a lot of us that, okay, you get one tall and one short. And the result is 50% of the tall plants and 50% of the short plants. So that's the beginning. And then he also do the two tall. He thought that, okay, if I have two tall plants, the result would that be only two tall plants? He found that no. He found that he got three tall plants, but about 25%, like 1/4 of the offsprings are short plants. Then why is that? So he's curious about it, and that led him to present or propose the very famous, established the very famous Mendelian genetics. And that's basically what we are a lot of our situations still apply to his law, his observation, not his law, he's observed the law, right? So it's not the law he creates or it's being there, he just observed it, okay? So that's that. So what we observed is, what he has observed, or we observe is the expressed that you can see. We call it a phenotype. So when we see a tall plant, a short plant, that's a phenotype. We see a red bird, a blue bird, that's a phenotype. And these different phenotypes are called a trait. We can have the trait to see a person's hair, whether that's a curve or it's straight, a trait of the phenotype that the plant is tall or short, or the flower is purple or yellow, or the eye color, right? And even persons tall, short could be a trait of the phenotype. Now, this phenotype is what you can observe. But we need to know that all these phenotypes come from as a result of our gene. So I am an Asian. It's not because I get to choose, right? It's because my parents are Asian, right? So if you have like Asian with American, like white, and the kid's gonna be a little bit here, a little bit there, right? So the phenomenon, the phenotype that you can see is because of the gene, it's because of the parents. And the parents give the offspring that phenomenon, that phenotype. It's because that we have, as we already learned from the cellular physiology, we already learned that that's the gene, you got one copy from the mother and the other copy from the father. And so you have two member of the same gene, one from mom, one from dad. And now that gene will be expressed to be shown and to be observed. So when we observe it, in the time when George Mendel observed it, he doesn't know about the gene, but he thinks that must be something from the parents, right? So it must be something, even though he has no proof, but the idea is already there. So it's a gene, right? So we have the observation, that's a phenotype, and the underlying source to show that phenotype is a gene. We call it a genotype. So different genotype will result in different phenotype. But as we mentioned before, even if it has the same phenotype, we could have the offspring with different phenotypes. So how can you explain the gene in these two which show the same phenotype, but the offspring apparently have different phenotype? Different phenotype must refer to a different gene, right? If it's coming from the gene, so it's short because it got a short gene. How can your parents are all tall, but you are short? How can you explain that? So that's the transition from the phenotype to the genotype. So it turns out that we already know that it's not exactly turned out, but we all know that each gene to express a phenotype coming from two members of the same gene, right? You have a hair gene from dad, hair gene from the mom. You have tall gene or short or length gene from the dad and length gene from the mom. So you have two copies, two members of the same gene from of the same gene, two copies, basically two copies of the same gene. So here we have a 23 chromosome. We have 23 pairs of the chromosome, right? So each gene can be kind of like a fighting to each other, like mom's gene, dad's gene say, he has to be tall, he has to be short, he has to be tall, he has to be short. So two genes kind of fight to each other, right? So, but you have two genes, right? You have two genes for the same thing. They have to fight. Someone will have to win to decide whether you're tall or you're short, right? Or to decide that your eye color gonna be blue or to be black, right? Like me, right? So that's that. This portion, this gene from two parents, gene from two sources, or gene of two copies are called allele. So say we have this gene, this is gene to decide the color. This could be, this same gene can have multiple copies of allele. In your body, you only have two copies, but in the society, it could be many copies, right? It could be, say, plants, right? It can be white flower, blue flower, pink flower. It's all here. And so you can have little a, small a, something like that, but same gene, but they're gonna have to fight with each other to decide whether this plant's gonna be the color of that or this. So that's the allele. So here is the genotype and the phenotype. We have the observation, that's the phenotype. The allele components in that body is called the genotype. So let's see how genotype defines the phenotype. So basically that in each gene, we have the two alleles. Let's say one from the egg, one from the sperm, right? And so we have, if these are two different alleles, right? It could be tall, it could be short. The combination could be several, right? The kids or in the society, in the plants that Mendel is looking into. Let me drink something first. George Mendel was looking into, it basically has two types, right? Tall or short. So it could be have gene that represent that tall one, a small a represent that short one. They are all A, right? These are all lengths. So in his garden, he can think that, or in the reality is that, he can think that this could be four possibility, right? It could be that these two are all capital A, or it could be capital A, small a, or it could be small a, capital A, or it could be two small a. So it could be, there's no other way. It could be only one of these, right? And now he observed one thing quite important, which is that when these two alleles are different, say this one is the same. So if the big A, capital A is tall, this plant is gonna be tall. If the small a is short, this plant is gonna be short. Now, if these two are different, this is called heterozygous. This is called monozygous. We will talk about that. So if these two are different, then they're gonna have to fight, right? They're gonna have to fight. One will win, okay? The one which win is called dominant. The one which lost is called recessive. So if two genes are the same, it's called the homozygous. If two alleles are different, it's called heterozygous. Zygous means that the fertilized egg, right? Zygote. And homozygous is that gene, that allele, that gene has the same allele in that chromosome. And the heterozygous is that gene has coming from different allele. So when different allele is in the body, these two will have to fight to show which one will win. And the one which win is called the dominant allele. Okay. So if you have a big, like a capital A, then you have to show capital A phenomena or phenotype of the capital A. If you have capital A, small a, then the phenotype is gonna be capital A, okay? Because that is the dominant allele. All right, so problem solved. So that basically is what George Mendel has observed, and that's what we are using right now, and which is quite usable. So here is the genotype versus phenotype. Say we have the red color is the dominant, is caused by the dominant allele, and the blue color is caused by the recessive allele. And so even though we have basically three different possibility of the combination from these two alleles. It could be A, capital A, capital A, capital A, small a, small a, small a. Even though we have three different possibilities, the phenotype is only two. If it's capital A is showing us the red color. If it's capital A, small a, it's still showing us a red color. So phenotype is one thing, but the underlying genotype could be different, but they are related. So that's the genotype and the phenotype. So in George Mendel's experiment, in his study, he looked into several phenotypes. When we look into different phenotypes, we call it phenotypic traits. So different traits. So say tall versus short, that's one trait. That's right here. That's right here, short versus tall. He also observed the flower position on the top, on the terminal, or on the middle. He also observed the color of the pot, the form of the pot, the color of the seed, and the form of the seed. So each one of these is a trait, and each trait is controlled by a group of alleles, and that including, for example, the gene capital R, small r, to define whether the seat is smooth or wrinkled, the tall or short, capital T, small t. Yeah. And by doing so, he observed that if the parents, right here, if the parents are all tall, but kids could be short, it means that the tall must be a dominant allele. Short is recessive because these two tall genes must carry one short allele. The one short allele is the reason we have the short kids. But the one short allele is not showing here because the short allele is a recessive allele. So here, if the two same phenomena provide the offspring that's different phenomena, that different phenomena must be a recessive. So that's his observation. And in our human being, we can also typically find different traits that can fit into Mendelian law. Mendelian law, yeah. So it could be that your parents, if you look at yourself, whether you have, it's called widow's peak, that's the hair right here. This is a dominant. If your parents are both have this peak, but you don't be surprised. You are still their kids, okay? So, because this is a recessive, if you don't have it, I don't have it because I don't have hair, okay? If you don't have it, it doesn't mean that you are not their kids. It's because that this is a recessive. Your parents must carry that recessive gene, recessive allele, but they don't show it because they also have the dominant one. And just by chance, one fourth will have the kids with the one we don't have it. Freckles is a dominant one. If you're the one without it, if your parents both have it, you are still their kids, okay? Tongue rolling. How am I doing? I think I can do it. Also the mid-digital hair. I don't have much hair, so I don't even have hair. All right. And also the ear lobes, right? It's really hard to say, maybe a little bit. And the thumb, right? And dimple in chin. I don't think I have it, but my kids have it. Very cute. I like it. Ah. Also the Morton's toe. All right, so that's different traits, okay? Different phenomena means have different traits. Different trait is control. It's represented by that two allele. They are fighting to show freckle or not. Tongue rolling or not, okay? So let's do a practice. Say if the parents are both, if you got the bird, you got the red birds, what's the chance you have the blue bird kid? What's the chance? Two possibility, right? You can have no blue bird chicks, For example, let me do that. Let me do that. Let me get it here. Let me find it. Okay. Right, I can do this one here. That. Indentation, that's this one here. So say we have a, say you have a red bird, right? So in here, if your red bird happens to be, okay, weird, big A and big A, okay? Then the chance of the kids to have bluebird is almost zero, right? Because you have AA, this one will give the, say this is female, it's gonna have one egg, one egg of big A, the other egg of big A. So you got one egg A, the other egg A, and here is a sperm, right? So here is a sperm. This one is another sperm. Yeah, so you can have a sperm here, small a, or another sperm here, big A, sorry, big A. So if they mate, these two mate together, gonna be AA. These mate together and the AA, right? So these two mate together, gonna be AA. Okay, so you have no chance to have a bluebird. Okay, so that's not. This will not have any bluebird. Clear all. Now, if like this one, okay, this one, you have one bird. So here is the female again, you got one egg. Ah! And the other egg, big A. This is sperm, one sperm, and the other sperm, small a. Now, you can have, if this sperm meet this egg, it's gonna be AA. If this sperm meet this egg, it's gonna be AA. If this sperm meet this egg, it's gonna be AA. If this sperm meet this egg, it's gonna be AA. So how many chance you will have the bluebird? Zero, right? Because it's all dominated. This one gonna be red, this one gonna be red, this one gonna be red, this one gonna be red. Okay. So in what situation you can have the bluebird as the baby if this one has big A and a small a, and this one has big A and a small a, then the egg could be big A, small a, right? And the sperm could be big A and small a. Now you will have chance that if this met with this one, it's gonna be AA. If this made this one, it's gonna be AA, right? If this made this one, it's gonna be AA. If this makes this one, it will be small AA. So you do have the chance. So it really depends on the genotype. But for the dominant allele, for a dominant phenotype to have a recessive phenotype, it's possible if parents are both heterozygous. If one parent is not heterozygous, then the chance is none. If the parents has both homozygous on the dominant, then the chance of getting a recessive phenotype is zero. Only if the parents are both heterozygous, like this one here. Only if the parents are heterozygous, then the chance to have a recessive is there. So to have the recessive phenomena is very difficult. I mean, the chance is very low. You must have the parents to have the recessive allele, and the both must have it, okay? So that's the real. Let's move on to the next one. Close this one. All right, okay. So, so the dominant trait will be happening successively from one generation to the next generation. But the recessive trait can be skipped, like the one we showed before. To have the recessive trait, it's basically skipped because their parents has no recessive trait, but their grandparents probably have it. So the trait will be skipped through this generation to the next one. So recessive traits can skip generations. So that's. Now let's talk about the autosomal trait. So when we talk about this autosomal, just use the quadrature. Just use the mouse here. So autosomal means that it's not from the sex gene, it's from the autosomal gene. And we have 23 pairs of the chromosome, 22 pairs are autosomal chromosome. So autosomal dominant traits can affect both male and female. Male to male transmissions can occur because from father to the son, right? It's possible. It doesn't really matter about the sex. When we talk about this practice, we don't really care whether this is mom or this is dad. We can be dad and mom, right? Doesn't really matter. So sex is not. Male, female transmission has equal frequency. Successive genes are successive generation are affected, and transmission stop after a generation in which no one is affected. So that's dominant. If say the dominant gene that produce kids with no dominant trait, then these kids must carry only the recessive, and they would the dominant allele would become, they will not be continued. So that's the same thing criteria for the autosomal. So that's for the recessive. The recessive is autosomal, means male, female are both affected. The only difference is that this trait can skip generation. Okay, so that's the difference. So to guess the frequency of one phenotype, the way to do it is to use this kind of the square. It's called Punnett square. We kind of do it before, right? So the parents have two allele, the other parent has two allele. Then we basically put them all together, and we can get all the possibility, all the choices of the offspring. And from here, we can quantify the frequency of one phenotype or one genotype to be expected from their offspring. So for example, here, the frequency to have the homozygous dominant alleles is 1/4. The frequency to have the heterozygous alleles are 2/4. So that's 1/2 is 50%. The frequency to have two homozygous recessive alleles is 1/4, so it's 25%. So from here, we can quantify the frequency of a genotype. 1/4 is the homozygous dominant alleles. 1/4 is the homozygous recessive alleles. We can simplify it like this one here. So that's how we do it. And this is the same idea that about 200 years ago, George Mendel has observed. So this is the genotype. And from here, we can have the frequency of the phenotype. The frequency of the dominant phenotype is 3/4, so it's three quarters. The frequency of the recessive phenotype is 1/4, is 25%. So we can have a practice that if these two tau genes is the tau genes result of a dominant allele or two recessive allele. So if we have this result that turn out have one recessive allele, the parent's allele must be heterozygous, okay? Must be heterozygous, must contain dominant allele as well as the recessive allele. If one is not heterozygous, if one is the homozygous, then we would not expect any recessive phenotypes in their offspring. And so we can do a practice that say what's the possibility of these parents. For example, this one, we can kind of know that their parents must be, even though we don't do gene study, right? What? We don't do gene study, but we can kind of, based on the experiment, we know the parents must have heterozygous, must have A, and A, because only then we have A, 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 we have the chance to have the recessive allele. And how about this one? So let's clear it out. How about this one? How about this one? What's their, so if we have the experiment like this, what's the genotype of the parents? What do you think? So we can try that, right? We can try that. We can have only two possibilities. One is that, so this one must be small a, right? Because this is recessive. This one could have two possibilities. One is AA, the other one is AA, right? So we can test this cross or this cross and to see the result. So if this is the one we are going to cross, then let's say A, do this one here, AA, right? And the result is A. Do we have any recessive offspring? No, so this is not the case. Then how about this one? This one, try A. So it's going to be A. We found that 4 kids, if we have 4 kids, 2 of them gonna be recessive, 2 of them gonna be dominant, 2 of them gonna be short, 2 of them will be tall. So that is the one. So from here we can know that that's this one. This one is the tall parents. This one is the short parents. So we can use this Punnett square to estimate based on the experimental findings to kind of like define the genotypes based on the experimental finding. So that's that. So we can test cross. Say we have tall one, we have short one. The tall one could be capital T, capital T, capital T, small t. So basically this is what we just did, right? So if this is what we guess, then the kids will have no short plants. If that's this one, then we have two short plants in 4. So about 50% tall, 50% short. So that's that. Now let's summarize it. The Mendel's first law is called gene segregation. And that law is presented as that equal allele distribution into gametes. So if the parents have 2 alleles, that will be equally distributed. Say if the parents, right, if say I have 2 alleles and they're gonna be distributed to the gametes. Gametes are all haploid. 50% of them will carry 1 allele, the other 50% will carry the other allele. So that will be 50-50. So that's one law. And then the second law is that when they mate, when they fertilize with another, the chance will be random. It could be the randomly combined. Chance will be equal, basically, will be randomly combined. So that's how we can use these square, because no one is more important. No one has higher chance than the other. Their chance will be the same. So that's the first law. The first law coming from the the parents, the gametes, the allele present in the gametes are has the equal chance to carry one of the allele, and their mating gonna be, their fertilization gonna be randomly picked. So that's the fertilization process can explain the Mendel's first law. So this one just showing you again that the dominant inheritance, basically that if the parents, even though that the parents is a heterozygous with a recessive allele, the result could be 50-50, right? And also that if the parents are dominant allele, dominant phenotype, then the kids too have a recessive phenotype. If that's happening, then their parents must both are all heterozygous. Say two tall kids, two tall parents, to have one short, to have a short kid, the parents must be both heterozygous. In this case, two parents are all both are curly hair. That's the dominant one. And turn out one kid, it has a straight hair. So the parents must both heterozygous. This both heterozygous cross is called monohybrid cross. Mono means the same. They are both the same phenotype. Hybrid means that each one of them are all have the heterozygous alleles. So monohybrid. Monohybrid is a way to describe that why parents don't carry that phenotype, but the kids do. And the kids do means that this phenotype must be a recessive one. And also parents are monohybrid cross. Okay, so that's that. Another thing you kind of need to be very familiar is that this phenotype, this recessive phenotype, is about 25% chance. Here is the Punnett square. So we can see that for the monohybrid cross, the genotype ratio is 1 to 2 to 1. One monocyte, one mono, so one homozygous, one homozygous dominant, two heterozygous, and one homozygous recessive. So 1 to 2 to 1. But the phenotype is 3 to 1. So the ratio of genotype and the phenotypes are different. The recessive chance, the chance to have a recessive phenotype is 25%. So that's that. So it basically, when you learn about this Mendelian law or genetics, a lot of time it's a terminology. You need to be very familiar with that terminology. Now that's what Mendel's first law. First law is the all alleles are equally distributed, and their mating are also randomly picked. Let me go back to there. Here, Mendel's first law. So all the alleles are equally distributed to the gametes, and their mating are also randomly picked. This is also called gene segregation. So basically that separation is equally applied. So that's first. And there is a second law. Second law refers to that two genes are independent to each other. Say, because here we have Mendel's experiment. He looked into different traits. There is R gene, Y gene, B gene, vis-a-vis G gene, right? Green versus yellow, tall gene, tall versus short. These, each one apply to the first law that they are equally distributed to the offspring and their mating is randomly picked. If you have two different traits, these two traits are independent, so they are not going to be interference with each other. So that's the second law, right here. So two genes of different chromosome means that they are not alleles. They are not homologous. They are so they will not interfere with each other. They will act independently. This is also called an independent assortment. So the first law is gene segregation. The second law is also called independent assortment. So that's the two main law of Mendel's law. Now you know it. So this one showing you that B gene, H gene, H allele, and the E allele, their combination will be independent to each other. So if you have parents have big A, big B, big H, small e, then the chance can be, if you want to do the phenotype frequency, then each one will be independently conducted. Okay. So that's that. So in summary, that's basically we describe all these different terminology, and this terminology basically represent an idea and a concept. So we learned about the phenotype. Phenotype is the one that we can observe, and that observation basically reflects what's going on in the gene. So different phenotype, we don't call it different phenotypes. We call it different traits. It could be the eyelid, it could be the hair, straight or curved, it could be the tall or short, different traits. And these different phenotypes are controlled by the genotype. So to know the person's allele, whether that's big A, small a, that is the genotype. We can do genotype by getting one cell from our body. A very easy one is to get a skin cell. So basically we can swap the skin cell in the mouth and just put it in the bag and send it to the company, and they will do the genotype for us. They will show you that you have the allele, right? You have, even though you thought that your hair is curved, you thought that you have the dominant curved hair, but maybe you have the recessive one. You just didn't know. You never know. And one way to know it is to have kids and turn out he or she has a straight hair. You will know, right? Or you can do the genotype to test whether you have the recessive gene or not. And the one gene has two allele, one from mom, one from dad. And these two allele can be homozygous or heterozygous. And the dominant one will be the one to show the phenotype. So that's that. And to know this terminology. So this is key concepts that we have learned, from the Mendel genetics. Heterozygous are two alleles have the same identical alleles. That's homozygous. If two alleles are different, it's heterozygous. The one who show the phenomena is called dominant allele or dominant phenotype. The one only show if they are homozygous is the recessive one. And if the parents are both dominant traits, but the kids do have the recessive trait, then both parents must be heterozygous. It's called the monohybrid cross. The genotype is 1 to 2 to 1, but the phenotype is 3 to 1, means that there's a chance to have the recessive trait, but the chance is 25%. And this also refers to a skip generation transmission, that the parents don't have the recessive trait, but the kids do have it. That means that the trait must come from the grandparents. And then we can use the Punnett squares to very easily quantify the frequency of the genotype and the phenotype. And using, if we don't get the gene test, we can know the genotype by using a test cross. If we cross it, have the kids, then if the kids number is big enough, meaning that if you only have one kid, you really didn't know the parents. But if you have 4 kids, then you have good number to know whether the parent, what's the genotype in their parents. And that's the way what George Mendel did. He did not do the gene test, but just by observe the offspring, he can know the parent's genotype and also to know the kid's genotype. So that's that. And the next question is about blood type. So this is very common that the two couples, how many slides do I have here? Let me check. Okay, we are halfway through. All right. So blood typing. We can do blood type, right? We can test the blood type. That you must know your blood type because that is quite important. Personal stuff, but quite important. So if the parents, if two couples have different blood type, what's their kids? So how can you define it? Can type A and the type A to have kids type O? If type A and type B to have kids type O? Yeah, so that's a question. All right, so we will answer it here. So before we do the Punnett test, we need to know what defines the blood typing. Blood typing is a type of the red blood cells. So in the red blood cells, even though that we all carry red blood cells, we have 4 types of red blood cells. It's because that red blood cells has its feature. We call it antigen. So for the blood type A, it carries the A on the cell. For the blood type B, it carries B antigen on the cell. For blood type AB, it carries type A and type B in the cell, on the cell. So this is basically caused by the enzyme, but don't worry about that. I should have removed that, but it's antigen. Antigen A, antigen B, antigen AB. And the type O has no antigen. So blood type A, it's possible that blood type A is caused by because the blood type A has the allele, right? It could be that both parents are type A, or it could be the blood type A or none. Because as long as you have the allele which can generate the antigen A, then you will be blood type A. So you can have the parents from type A and type A, or from the parents type A and type O, you will have type A. You have the chance to have type A. Same thing as the type B, it could be the allele, both allele are both B, or could be just one allele B. If you have one B, you have one gene to express that enzyme to produce the type B antigen. For type AB, you must have type A and type B, because if you only have A and A, it's gonna be A. If you only have B and B, it will be B. If you have A and I, means no antigen, it will be A. If you have B and I, it will be B. So it must have the A and the B. And the A and the B don't fight. It's not like one will win the other. It will be both show. So that's the amazing part of the blood type. In tall and short, you have to fight. You cannot be both tall and short. For the wrinkles, right? Sorry, the freckles, yeah, freckles, right? For the freckles, you either have it or not. It's not like both, right? Yes and no. So, but A and B is very unique. Is that if you have type A and type B allele, they won't fight. Each one will generate its own enzyme, and each one will generate its own antigen. In a way, it's kind of independent. And both will show, even though they both are located in the same gene, in the same location, and the different allele will just do their job, and they will both show. So they don't fight. So you will have the type AB, and that both different allele that are both expressed. That's the case like this one. It's called co-dominant. Both are dominant. No one is recessive. Both are dominant and both are shown. That's type AB. And type O is one who doesn't carry any of these A and B alleles. So that's that. Now, with that in mind, it will be easy, right? Say the question was, can type A have kids? Let's see the question here. What's the question? If the couple have blood type A and blood type B respectively, what's the blood type of their kids? So it could be the I, or it's right here, okay? It could be the A and B, right? It could be the parents are both AA and the BB. Then their kids will all be AB, 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 AB. If the parent is only one allele carry type A, the other allele doesn't, then their kids will be A, B, AB, and IB will be BB if the parents is both AA and BI, the kids will be AB, AB, AI. AI will be AB, type AB, type AB, and type A, and type A. If the parents are AI, BI, it's still type A, type B, but the kids will be AB, BI, AI, and II. II will be O. So in this case, type AB, type A married type B, the kids will have AB, A, B, and O. Okay, everybody has one chance. Okay, so that's that. So that's the broad typing. Now, another, I don't know why this one is not working, but okay, right. So another concept, another terminology is the pedigree chart. Pedigree chart is a chart to show the family tree, okay? So the diagram to show the occurrence of the phenotype from one generation to the next. So it's basically a family tree. And that is like this one here. So here are just symbols that white means normal. Black usually refer to disease, okay? The trait, okay, trait. And each person can have both white or both black because it has two allele. And then round is female, square is male. So if it's heterozygous, it's gonna be half black, half white. Cross is the dead. If the sex doesn't know, you use this shape. P is the one still not delivered yet, still pregnancy. Abortion, spontaneous abortion, miscarriage will be, or terminated pregnancy will be that. And the last line is generation to generation, parents, partners. And so let's take an example, take a look. For example, this one. So this one refers to the pedigree chart of the albinism. Albinism is a recessive. It's an autosomal recessive, means that it affects both sex. It's not it's sex dependent, independent, and it's recessive. So say if the parent's mom has one recessive gene, the dad doesn't, it's not albinism. The chance is that their kids would not, because still have the recessive gene, but it's not showing, so it's not albinism. And if he married to another person who also coming from this tree and carry the albinism recessive gene, then they do have the chance to have the recessive homozygous in their kids. This one also show you that if the marriage is in the relatives, meaning that if this recessive gene has higher concentration in this family already, it's very likely that recessive gene gonna become visible, become the phenotype in their kids. And sometimes these recessive genes are not good. That's the reason why we don't show it that often, because if it's become visible, then they probably won't have the offspring. The recessive gene remain in the population is because they are recessive. It's because that they don't show, and they are not dominant. They don't affect the health. If it become the homozygous, then it will affect the health, and this will be sick, and this will be they will not have the offspring. So this is chart of the pedigree chart, and the term to describe that marriage between relatives is called the consanguinity. Consanguinity. And that is quite common in the old time in this royal family, that they marry with their relatives, and those recessive genes will become highly concentrated in a population, and that will cause the chance to express that recessive phenotype quite easily in the family. Is that it? All right, what else? Yeah, we do have some more. So the next topic, the next portion is called the beyond Mendelian genetics. Beyond Mendel's law. Okay, so what's that? It's a concept that in Mendel's law we have the idea about the two genes fight, right, and the one dominant the other one, but we see this in some traits, but not all traits. It's not always the case. For example, so this leads to the concept, these two concepts called penetrance and expressivity. Penetrance and expressivity. It means that how far this gene will affect its offspring. Expressivity is that how severe this is. For example, we see the tall allele, but how far it makes this person tall. It could be middle tall, it could be a little bit taller, or it could be not tall at all. So that's the expressivity. Penetrance is that how does it really cause it? For example, this is commonly seen in the disease gene, like cancer gene. This gene gonna produce the enzyme to cause cancer, but it might not happen, right? The very common one is the Alzheimer's disease gene, that the parents carry this gene to produce this amyloid peptide, that's the bad protein in the brain to develop Alzheimer's disease. But this might not happen. Say both parents have it, both parents have frontotemporal dementia, that's also a mutant cause that disease. But their kids, even though still has that mutant, they might not have it. So that's the penetrance. Because the reason is that they may not express. You know, you have the gene to express the bad protein, but that gene might not express. So the penetrance, and that will make the Mendel's law, you know, not work, because we thought that the kids, the parents both have the dominant gene, the kids will have it, but turns out they probably won't have it. They probably can affect it by the diet. they probably can suppress or not showing that gene, not express that gene by the diet, by good lifestyle, by, so many different things can affect it because gene not work independently. They interact to each other. All right, so the penetrance refers to all or none. So it's usually the disease like Alzheimer, tumor. You don't have middle Alzheimer, you have it or not. You have a cancer, you have it or not, right? So it refers to the one that's yes or no. It's not like a middle. Tall or short, we can have the middle, but in some feature, it's yes or no. You curve hair, yes or no, right? The genotype is incompletely penetrant if some individuals do not express the phenotype, okay? Even though it carry it, but do not express. So it's called the incompletely penetrance or incomplete penetrance. In reality, the completely penetrant is very rare. So, for example, breast cancer is almost like a #1 cause of death in female, very high. Cancer is already very high. It's the top cause of death in America. And breast cancer is very high for female death, to cause the death in female. So, and there are some genes show very high chance to develop the breast cancer. And but the thing is that they might not show, it's not necessary. There are so many things can affect it. For example, Angelina Jolie, right? Her parents, her mom, her relatives, both all have the breast cancer, and she has that gene too. So she decided to remove her breast. I remember so. Maybe I was wrong. So that's one way. If you don't have the breast, then it will not show. But even though you have that gene, it might not show. It's not necessary. It's not 100%. Even though there's chance, it's not always the case. If 80 of the 100% people, if 80 of 100 people who inherit the dominant polydactyly allele, means that it has the group. It's not just one gene. It has group of that dominate. If 80 show of 100, then the penetrant is 80%. So that's the penetrance. Expressivity means the level, the severity, or the level of that gene to be the intensity of that phenotype. For example, some people has the same asthma genotype, but the severity could be different. How our immune system responds to the allergen could be high and low. There are so many things can affect it. How many receptor in the cell to detect that allergen, and how is your immune system respond to it? You will cough more, you will sneeze more, you are more sensitive, you are less sensitive. It could be different. So that's something that's more complicated, even though the Mendel's law still hold, but in reality, this is more complicated. There are many traits that could be complicated. So here is the homework and assignment that you will be doing. Say twins, monozygous twins, have the exact same gene, but would they have the same life? Are their fate is already determined, could they live a different life? One develop the diabetes and the other one would need to worry about it, or would he or she would not develop the diabetes? What do you think? Is that possible? It is possible, right? And your reason? What's the reason? Do you have personal observation related to similar phenomena? So I want you to think about it, do a little bit research, do a little bit observation. If you see a family that everybody are diabetes or everybody is obesity, but one kid doesn't, or the whole, you see your neighbors or you see your friends, you see some phenotype, but do you see something different there? Or if you really know someone with identical twins, do they develop different kind of the traits, different kind of phenotype? Yeah, so this is the assignment. It would be total 20 points, and you post it, your observation, and you reply to others, that's 5 points. You reply to the other, that's 5 points. So total is 20 points. So that's the discussion, that's the assignment. Another reason that Mendelian law probably won't, Mendel's law probably won't work is because some allele are lethal. So the kids carried that allele die or did not develop. The zygote won't develop, so they will not be observed, right? So there's no chance to become a living organism to be to show that phenotype. They probably just don't show because of that zygotes won't develop. This usually happens to the recessive gene, the dominant one, less common. One example is the hairless dog. This gene is a dominant, so the hairless dog are all heterozygous to mate. All of them are heterozygous, and the homozygous dominant won't survive. There's just too much of the hairless, right? It could be the reason. Another reason that Mendel's law won't work is that genes, in fact, are not independent to each other. In Mendel's law, each trait will be considered independent to each other. That's Mendel's second law, right? But the reality is that genes won't act independently. Our body, each gene will produce proteins, and proteins will have its function, and their functions are all interactive with each other. So it's not isolated world. They are flowing around, they will interfere with each other. Our life, our body is a dynamic organism. Different proteins interfere with each other. And so we have, this is called the gene interaction with each other. This phenomena is called the epistasis. And we have the gene called the modifier gene. For example, the hairless gene will affect the hair color, It's so obvious, right? If you like, if the person is bald, there's no chance to know that it's curved or not, right? So that's the basically Mendele's second law is difficult to hold because genes don't work actually independently. Some genes may work independently, but a lot of genes actually the expression actually interfere with each other. Another reason for the Mendele's law won't work is that some of the disease is caused by the mitochondria DNA. Mitochondria also has the DNA. That's why we have the earlier segment to talk about the cell. We need to have the background about all these different organelles. Mitochondria DNA only coming from the mother, so the disease is not equally distributed. It's not sex independent, it's sex dependent. It's coming from the mother. And also that in one cell has only one nucleus. So we have one copy of this allele. But in the cell, we have many mitochondria. Each mitochondria can have different DNA. Okay, so you may have a disease DNA, a mutant in one mitochondria, but the distribution could be varied. One egg may have a lot of bad mitochondria, but the other egg might only have a few. So that distribution could be different, and it really depends on if you're lucky, the offspring don't carry that bad mitochondria at all. So even though mother has it, the kids might not have it. Mitochondria gene also highly exposure to oxygen free radical means that they are easily get mutated. Mitochondria gene has about 33 genes. Mitochondria DNA has about 33 genes. They are easily got mutated, and so they do have higher chance to develop abnormal some mutants. So this leads to another idea. It's called the heteroplasmy. It means that each egg, each oocyte contains the mitochondria, but the percentage can be different. You know, some cells, if we have bad mitochondria, right? Maybe one egg carry a lot of it, but the other egg doesn't. So that distribution could be different. And that's the expressivity will be vary among the siblings, because some may got the egg which carry a lot of bad mitochondria, but the other one may got the egg with no bad mitochondria at all. So this one showing you the picture that if the oocytes has some mitochondria with mutations, that's the purple one, but the purple one distribution can be varied. Some egg may have only one, the other mitochondria are healthy, or it could be a lot. So, and the degree of the phenotype severity could be different. I think that's the last slide, right? Let me check. Yeah, that's the last slide. So that's about that's all, you know, Mendelian genetics. Let me know if you have any question, okay?